Alrighty, welcome back. Last little bit of chapter 8 here. The last section here is going to be on how the number and structure of our chromosomes can be sort of changed, sometimes in maladaptive ways, during the process of mitosis and meiosis. So whenever you have sort of an abnormal chromosome number, right? Remember, for humans, it's 46. That's the normal number. But sometimes humans ends up with extra chromosomes or not enough chromosomes. Anytime you have this sort of abnormal number of chromosomes, it results from what we call a non-disjunction. And there are several ways that non-disjunctions can occur. First, it might involve either the failure of a pair of homologous chromosomes to separate during meiosis 1, or the failure of a pair of, the failure of the sister chromatids to separate during meiosis 2. So here is our non-disjunction occurring in meiosis 1. So the cells still separate normally, but these homologous chromosomes didn't separate. And so even though meiosis 2 is completely normal, these gametes end up with extra chromosomes. These gametes end up with not enough chromosomes. And so you end up with these very abnormal sex cells. Here we have a normal meiosis 1. The pairs of homologous chromosomes separate like normal. The cell splits like normal. But then during meiosis 2, a non-disjunction occurs. One of these pairs of sister chromatids don't separate. So here, since the disjunction only happened in one of the meiosis 2 cells, these gametes are normal. They've got their normal haploid number. But here you have an abnormal gamete that has an extra chromosome, and a normal abnormal gamete doesn't have enough chromosomes. And so whether it's a failure of a separation of the homologous chromosomes or the sister chromatids, either way we can end up with sort of an abnormal number. How do we determine if somebody has an abnormal number? Typically what we do is we make what is called a karyotype, which is sort of this photographic kind of look at a person's chromosomes. Typically what you do is you'll take some blood from the patient, you isolate those white blood cells, spin them in a centrifuge, separate out the red cells from the white cells. You add a little fixative, you add a little stain, you get those cells to grow, but you stop them right at that metaphase when they're all lined up. And then you use a microscope to photograph them, typically sort of in ordered pairs, and by arranging them in this very specific pattern, we can look at if there are any abnormalities present in either the number or structure of the chromosomes. So here you can see, here's our sister chromatids, here's our pairs of homologous chromosomes, and you can see here's the karyotype. And you can see here's chromosome 1, 2, so on and so forth. And here are our sex chromosomes, so XY, right? That determines your sex, whether you are male or female. If you know anyone with Down syndrome, for instance, I have a niece who has Down syndrome. It's called trisomy 21. Essentially, they've got an extra chromosome here at chromosome 21. So instead of having 2, they've got an extra there. Some Down syndrome people are, have pretty distinctive features in terms of some of their facial features, but sometimes there are developmental delays. Sometimes there are sort of on a spectrum, kind of like a gradient, sort of like autism. Some cases of Down syndrome are more severe than others. They've actually made great strides recently in caring, just in the last 20, 30 years even, caring for people who have trisomy 21. 20, 30, 40 years ago, if you had a Down syndrome diagnosis, the expected lifespan of somebody with Down syndrome was pretty young, in the early 20s, but now there are adults who have Down syndrome that still live, very fulfilling lives. So it's not nearly the, shall we say, fatal problem that it once was, which is sort of amazing. I love when we make progress, right, guys? Progress. For instance, Down syndrome people, or let me put it this way, your risk of having a Down syndrome child tends to increase with the maternal age of the mother. So if you're, young and in your 20s and having kids, your risk of having a Down syndrome child is probably pretty low. But if you're, say above 45, then, that greatly increases. In part just because those meiosis, nothing works as well when everything is a little less efficient when you're older, right? Not that 45 is that old, but in terms of reproductive health, it kind of is. So sometimes an abnormal chromosome number can be fatal. Trisomy 21 is not. So that extra chromosome at number 21 is not fatal. There are some chromosome abnormalities that are fatal, which would result in a miscarriage. But if you have non-disjunction in your sex chromosomes during meiosis, sometimes what you get is missing or extra X or Y chromosomes. Now, sometimes that's can affect the person's health, and then sometimes if you, for instance, if you're female and you have an extra female chromosome, it's really fine. It's totally normal. So you can see, sort of depending on where the disjunction occurs, sometimes you might be pretty normal, sometimes you might not be normal, sometimes there might be just slight variations, so kind of an interesting thing when happens when you get an extra sex chromosome. And the really just amazing thing that happens in terms of sort of mistakes, shall we say, in cell division, is that you can get what are called polyploid organisms, organisms that have sort of these extra set of chromosomes. And when you have a polyploid organism like this tree frog here, which is a tetraploid organism, you get sort of this interesting ability to evolve into new species. And so as with everything else in terms of biology, overarching all of this is the evolution of our species and our planet. Now, of course, there are ways that we can sort of mess up our chromosomes and structurally that can end up resulting in birth defects and cancer. So something like a deletion or a duplication, inversion, translocations, they can result in a whole host of different genetic disorders, or if they occur in a somatic cell, sometimes cancer. So for instance, a deletion occurs when a particular locus and a particular gene is just sort of transcribed out. It's just not included, so they're lacking some trait. Duplication is when the chromosome is sort of doubled, so it's extra. That can affect things and lead to some genetic disorders. Inversions, where things sort of get swapped and split, sort of that can result in disorders where you've got things not functioning properly. Reciprocal translocation, so here you have some non-homologous chromosomes sort of swapping genes, that can result in genetic disorders. Or if you had, for instance, reciprocal translocation occurring, here you have chromosome 9 switching some information with chromosome 22, and that can sort of activate a cancer-causing gene, which can then result in cancer occurring in some of your somatic body cells. So we do have to be careful about our chromosome structure as well as our chromosome number. So in terms of sort of summarizing the chapter, you want to be able to compare, contrast asexual and sexual reproduction, kind of explain why cellular division is necessary, the whole cell theory again, explain the process of binary fission, compare the structure and function of the eukaryotic chromosomes versus the prokaryotic chromosomes, describe the stages of the cell cycle, describe mitosis and the events of mitosis in each of the phases of mitosis, compare cytokinesis in plant and animal cells, talk about density, anchoring, growth factors, control cell division, cancer cells and their sort of exponential growth, the functions for the mitotic processes, how chromosomes get paired up, distinguishing between our somatic or body cells and the sex cells or the gametes, whether they are diploid, full chromosome number, or haploid, half chromosome number, why sexual reproduction requires meiosis, explain meiosis 1 and meiosis 2, what goes on in each of those phases, compare and contrast mitosis and meiosis. Compare contrast questions are always good test questions. Genetic variation in terms of sort of independent random assortment and also that chiasmata, the crossing over. Karyotyping, how we do it, sort of why we do it. A little bit there about Down syndrome. Abnormal sex chromosomes. Explain disjunctions, what can happen as a result, and how sometimes new species can evolve from errors in cell division, and sort of how chromosomes get changed in terms of those deletions and insertions and so on and so forth. So that is chapter 8.